So if you step into a functional medicine clinic today, or, maybe you're just scrolling through the social media feed of some high-performance athlete, you might see something visually jarring.
Oh, definitely.
Yeah.
It's hard to miss.
Right. Someone opens their mouth and their tongue is stained this vibrant, glowing blue, like they've just eaten a handful of melted crayons.
Yet that's a pretty accurate description, honestly.
But I mean, they aren't doing this for shock value. They are actively using a compound that sits at the absolute frontier of cognitive enhancement, cellular energy optimization, and, well, longevity.
Right, which is wild when you consider the flip side of it.
Exactly. Okay, let's unpack this, because this exact same substance is formally FDA approved to treat a highly specific, life-threatening blood disorder in emergency rooms. We are talking about methylene blue.
Yeah, it really is a striking visual, and that duality is what makes it so compelling, On one hand, you have biohackers using it off-label to theoretically upgrade their mitochondrial function.
Right, for the cognitive edge.
Exactly. But then, on the other hand, you have critical care physicians using it to literally pull patients back from the brink of cellular suffocation.
Wow.
And the mechanism underlying both of these applications is, well, it's entirely rooted in how this molecule interacts with oxygen and energy transfer at a microscopic level. It alters the fundamental chemical states of our cells.
And to understand how we got to a point where, a vibrant blue liquid is being used to theoretically sharpen the mind and delay cellular aging, we have to rewind the clock significantly.
Please now, way back.
We aren't starting in a modern sterile laboratory with some multi-million-dollar biotech startup. We are going back to 1876.
Right in the middle of the Industrial Revolution.
Exactly. Chemistry as a modern discipline was just finding its footing, right? And a German chemist named Heinrich Caro synthesized methylene blue. But he didn't create it as a medicine at all.
No, not at all.
He created it as a textile dye, like for clothes.
What's fascinating here is that historical context, In the mid-19th century, the chemical industry was primarily driven by the textile boom.
Right. People wanted colorful fabrics.
Exactly. And before this era, dyes were natural. They were extracted from plants or insects or minerals. So they were expensive, and honestly, they faded quickly.
I can imagine.
So chemists began experimenting with coal tar, which was this byproduct of industrial coal gas production, to synthesize artificial colors.
Yeah.
And methylene blue, which is a thiazine dye, was one of these early synthetic triumphs.
Because it held its color so well?
Yeah, it was brilliant, it was color fast, and crucially, it was cheap to produce. But the leap from a fabric dye to the very first synthetic drug ever used in medicine, that happened because of early microbiology.
Right, because if you're an early microbiologist and you're looking at a tissue sample under a primitive microscope, everything is just mostly transparent.
Yeah, it's just a clear blob.
You can't differentiate the structures at all. So they started using these newly invented textile dyes to literally stain the samples.
Exactly. And they noticed that certain dyes had a really specific affinity for certain biological structures.
Like they didn't just dye everything blue.
Right. Methylene blue didn't just wash over a cell evenly. It's selectively bound to specific components, like the nucleus or to specific pathogens.
Oh, wow.
And this selective binding was a total revelation. It led this brilliant scientist named Paul Ehrlich to develop the concept of the magic bullet.
The magic bullet.
Yeah, the idea that if a chemical could selectively bind to a disease-causing organism without binding to the surrounding human tissue, it could theoretically deliver a targeted lethal blow to just the pathogen.
That's the foundation of modern pharmacology, right?
Right.
Which brings us to malaria.
Right.
Because before methylene blue, malaria was treated primarily with quinine, which was derived from the bark of the cinchona tree.
Which was effective, but it was a natural extract.
Yeah. And then in 1891, Ehrlich and his colleague, Paul Gutman, realized that methylene blue specifically stained the Plasmodium parasite, the organism that actually causes malaria.
And they thought, well, if it targets the parasite so specifically just to stain it, what if we just inject the dye straight into the patient?
Which, you have to admit, was a massive leap of faith.
Seriously? I mean, injecting fabric dye into a person.
But it worked. Methylene blue became the first fully synthetic compound used as an active pharmaceutical in human history.
That is just wild to me. It's like an actor who started out doing silent films in the 1800s and is now somehow starring in cutting-edge CGI blockbusters today.
That's a great way to put it.
How does a fabric dye become the first synthetic medical marvel? What does that tell us about early scientific discovery?
Well, it tells us that they were highly observant. It successfully treated malaria by disrupting the parasite's metabolism. And over the next few decades, because of its really unique chemical properties, its application expanded rapidly.
Where else did they use it?
It was utilized in early psychiatry, it was used in neurology, and it just became an indispensable tool in laboratory chemistry.
But the true power of the molecule wasn't just that it colored cells. The color change was actually a visual indicator of a much deeper chemical reaction occurring within the cell.
Right.
Specifically, a transfer of electrons.
Which perfectly bridges us to how it is used in conventional medicine today, right?
Right.
Because it transitioned from this general antimicrobial into a highly specialized, FDA-approved blood mechanic.
Exactly.
And this all hinges on a condition called methyloglobinemia. To grasp what methylene blue does to our cellular machinery, I mean, we really have to thoroughly understand this condition.
If we connect this to the bigger picture, methyloglobinemia is a disorder of the red blood cells, specifically involving hemoglobin.
Stuff that carries the oxygen.
Right. Hemoglobin is the complex protein structure inside your red blood cells that transports oxygen from your lungs to your peripheral tissues.
Okay.
And at the absolute center of this hemoglobin protein is an iron ion. That iron is the literal binding site for the oxygen molecule.
And the chemical state of that iron is the whole ballgame here, because under normal, healthy conditions, that iron is in a state called ferrous iron, right?
Yes.
Which chemically means it's an iron ion with a +2 charge.
Correct. Ferrous iron, or Fe2 plus Na, has the perfect electromagnetic properties to bind reversibly to oxygen.
Reversibly being the key word there.
Exactly. It needs to grab the oxygen tightly enough to carry it through the turbulent arterial system, but it has to hold it loosely enough to let go of it once it reaches a low oxygen environment, like a contracting muscle or a firing neuron.
So ferrous iron is essentially an electromagnet.
That's a good analogy.
The red blood cell turns the magnet on in the lungs, picks up the oxygen cargo, travels down to the brain, and turns the magnet off to drop the cargo.
Precisely.
But in methyloglobinemia, something goes wrong with the iron. It oxidizes, it loses an electron, and transforms into ferric iron, which has a +3 charge.
And when it shifts to that ferric state, the physical shape and the electromagnetic affinity of the hemoglobin change drastically.
It gets stronger.
The ferric iron binds to oxygen with an incredibly high affinity. It refuses to let go.
So if a ferrous iron is an electromagnet that can be turned off, ferric iron is like a permanent rare earth magnet.
Exactly.
It picks up the oxygen in the lungs, but when the red blood cell arrives at the brain or the heart, the bond is just too strong. The oxygen is locked to the hemoglobin.
Right.
So going back to my cargo analogy, you have a delivery truck full of critical supplies parked right outside the factory, but the doors are literally welded shut.
That is exactly what is happening, and the physiological consequence of that is devastating.
I can imagine.
A patient with severe methyloglobinemia will present in the emergency room with chocolate brown blood and a blue tint to their skin and lips.
Which is called cyanosis, right?
Yes, cyanosis. And you could put an oxygen mask on them and flood their lungs with 100% pure oxygen, and it will not matter at all.
Because the delivery truck is still locked, does this mean without this specific conversion, we are essentially suffocating at a cellular level despite breathing normally?
Yes, exactly. They are experiencing profound cellular hypoxia. Their tissues are suffocating in the presence of abundant oxygen because the delivery mechanism is fundamentally broken.
Wow. Okay, so the emergency physician administers intravenous methylene blue. What exactly is happening chemically the moment that blue liquid enters the bloodstream? How does it, unweld the doors of the delivery truck?
Methylene blue essentially hijacks and supercharges the body's natural repair system.
Okay.
Under normal circumstances, our bodies constantly produce tiny amounts of methemoglobin. But we have an enzyme called cytochrome B5 reductase that fixes it.
It reduces it back to the normal ferrous state.
Right. But in a methemoglobemia crisis, whether that's genetic or triggered by exposure to certain drugs, anesthetics, or toxins, that natural enzyme pathway is completely overwhelmed.
The system is flooded and the natural repair crew just can't keep up.
Exactly. When methylene blue is introduced, it interacts with a completely different enzyme pathway in the red blood cell, involving an energy molecule called NADPH.
Okay, I've heard of NADPH.
The methylene blue acts as an electron acceptor. It pulls electrons from NADPH, temporarily changing its own chemical structure into a colorless form called leukomethylene blue.
It actually loses its blue color.
Yes, temporarily. And this leukomethylene blue is highly reactive and immediately donates that electron directly to the stuck ferric iron.
So it hands the missing electron back to the iron, reverting it from the +3 ferric state back to the healthy +2 ferrous state.
Precisely. And in doing so, it restores the hemoglobin's ability to release oxygen. The delivery truck doors are instantly opened.
And the oxygen floods into the starving tissues.
Yes, it is a rapid, life-saving chemical conversion.
That is, that establishes the immense power of this molecule in the bloodstream. It's a highly effective, rapid electron transporter.
Right.
But, the reason athletes and biohackers are walking around with blue tongues isn't because they have a rare blood emergency in the middle of a podcast or a workout.
No, definitely not.
It's because of what happens when methylene blue leaves the general circulation and targets the central nervous system.
Right. And to affect the central nervous system, any compound first faces a massive physiological hurdle.
The blood-brain barrier.
Yes, the blood-brain barrier.
But the blood-brain barrier is fascinating because we often talk about it in the abstract, but structurally, it is a highly evolved fortress.
Oh, absolutely.
Like, the blood vessels in the rest of your body are somewhat porous, right? They allow nutrients and molecules to pass through the vessel walls into the surrounding tissue relatively easily.
Right, they have to feed the tissues.
But the blood vessels feeding the brain are lined with endothelial cells that are packed incredibly tightly together, what are known as tight junctions.
Yes, tight junctions. And beyond just the tight junctions, those blood vessels are wrapped in the end feet of astrocytes, which are specialized brain cells that add another layer of security.
Here's where it gets really interesting. It's like the blood-brain barrier is an exclusive VIP club with the world's strictest bouncer.
That's a perfect analogy.
Okay.
Because this barrier exists because the brain is highly sensitive to chemical fluctuations.
It needs absolute stability.
Exactly. A slight spike in specific amino acids, ions, or toxins that the liver could easily process that would cause catastrophic misfiring of neurons if it was allowed into the brain tissue.
Right.
Therefore, the blood-brain barrier acts as a rigorous filter.
So what determines if a molecule can cross this barrier? It's not just a matter of size, right? Is methylene blue just tiny?
Size is a factor. Definitely, large molecules generally cannot pass. But the most critical factor is lipid solubility, or lipophilicity.
Lipid meaning fat.
Yes. The cell membranes that make up the blood-brain barrier are essentially made of fat. Water-soluble or hydrophilic molecules cannot easily dissolve through this fat layer.
Okay, so they just bounce off.
Pretty much. They require specialized transport proteins to physically escort them across. Methylene blue, however, is a highly lipophilic, low molecular weight compound.
Oh, so it just dissolves right through the lipid membrane. It doesn't need a transport protein. It bypasses the security checkpoint entirely. Methylene blue is literally on the guest list for the VIP club.
Exactly. It readily crosses the blood-brain barrier and accumulates in the central nervous system. And once it's inside the brain tissue, we start seeing its nootropic effects.
Nootropic being a term for any substance that enhances cognitive function.
Right. Studies have shown improvements in memory consolidation, enhanced focus, and an upregulation of executive function.
And executive function, for people listening, that's the higher order cognitive processes, right? Like planning your day, decision-making, and error correction.
Exactly.
Now, this is where I feel like we need to draw a very sharp line between a true nootropic and just a basic stimulant.
That is a crucial distinction.
Because if someone drinks 3 cups of coffee or takes a pharmaceutical amphetamine, they will also report enhanced focus and energy, right? But the underlying neurochemical mechanism is completely different. Like, is methylene blue acting like a massive espresso shot that just masks fatigue? Or is it fundamentally altering how our brain computes?
It's fundamentally altering the baseline. A standard stimulant primarily operates on the dopaminergic and noradrenergic systems.
So dopamine and adrenaline.
Exactly. It binds to receptors and triggers a massive release of neurotransmitters, or it blocks their reuptake. It creates an artificial state of high alert.
You're forcing the engine to rev.
Yes, you are forcing the neurons to fire more rapidly and aggressively.
Oh.
And the inevitable result of this is a crash.
Right, because what goes up must come down.
Because you deplete the stored neurotransmitters and you downregulate the receptors.
Yeah.
You haven't improved the health of the brain at all. You've just whipped it to run faster.
You've extracted future energy to use right now. You're borrowing against tomorrow.
That is exactly it. A true nootropic, on the other hand, alters the baseline metabolic capacity of the brain.
Okay.
It doesn't just squeeze more neurotransmitters out of the neuron, it improves the environment in which the neuron operates.
Wow.
And to understand how methylene blue achieves this, we have to look past the neural tissue, past the individual cells, and look directly at the mitochondrial matrix.
The mitochondria, the powerhouse of the cell.
Exactly.
This is the absolute core of the biohacking appeal. Because the fundamental reality of human biology is that the food we eat, the carbohydrates, the fats, the proteins, that is not the direct fuel that powers a neuron firing or a muscle contracting.
No, it's not.
All of those macronutrients have to be broken down and converted into a singular universal energy currency, adenosine triphosphate, or ATP.
Right. If you imagine your cells as complex pieces of machinery, ATP is the electricity.
Okay.
Without it, everything stops immediately. And the brain is the most energy-demanding organ in the human body.
Because it's constantly firing.
Exactly. It accounts for about 2% of your total body weight, but it consumes roughly 20% of your entire ATP production.
That's a huge ratio.
It is. When neurons fire, when they establish new synaptic connections for memory, they're burning through massive amounts of ATP. If the supply of ATP drops even slightly, cognitive function, focus, memory, clarity degrades proportionally.
So what does this all mean? If food, water, and air are like crude oil, then ATP is the refined gasoline the engine actually runs on.
That's a great way to visualize it.
So ATP is manufactured inside the mitochondria through a process called oxidative phosphorylation.
Yes.
And this happens along something called the electron transport chain. This is where I feel like the deep biochemistry is really necessary.
It is.
How exactly does this chain work, and where does methylene blue insert itself into the process? Because earlier we talked about it handing off an electron to iron in the blood. Is it doing something similar here?
It is entirely about electrons. So, the electron transport chain is located on the inner membrane of the mitochondria. You can visualize it as a series of 4 massive protein complexes, numbered complex I through complex 4.
Okay, 1, 2, 3, 4.
Right. When your body breaks down food, it pulls high-energy electrons from that food. These electrons are handed to complex 1.
Okay.
From there, they are passed down the chain from complex 1 to complex 2 to complex 3 and finally to complex 4.
It's like a microscopic bucket brigade passing electrons down the line.
Precisely. And as these electrons are passed from complex to complex, they release a tiny bit of energy. The mitochondria use that energy to pump protons across the membrane, creating a massive electrochemical gradient.
Kind of similar to water building up behind a dam.
Exactly like water behind a dam. Finally, that built-up pressure is released through a turbine called ATP synthase, and the spinning of that turbine is what physically manufactures the ATP.
Wow. And where does oxygen fit into this?
At the very end of the line, at complex 4, the electrons are finally handed off to oxygen, which combines with hydrogen to form water.
Oh.
That is literally why we breathe oxygen. It is the final garbage collector for the electrons at the end of the chain.
That is an incredibly complex, delicate system. It requires the electrons to flow smoothly through all 4 complexes without stopping. But the biological reality is that this system is not perfect. It experiences wear and tear.
This raises an important question. What happens when the system degrades? As we age, or when we are exposed to toxins, stress, or poor diet, these complexes, especially complex I and complex 3, become damaged.
They start breaking down.
They become inefficient. The electrons don't flow smoothly, they bottleneck. And when the flow bottlenecks, ATP production drops.
Ah, so the dam loses
Yeah.
pressure.
The brain has less energy to work with, which manifests as brain fog, fatigue, and general cognitive decline.
And this is the magic trick of methylene blue, isn't it? Because it doesn't just provide more raw materials to the mitochondria, it acts as an alternative electron carrier.
This is its most profound mechanism. Methylene blue has the unique chemical ability to accept electrons directly from the start of the chain and physically shuttle them all the way down, donating them directly to cytochrome C, which feeds complex 4.
So it's acting like a shunt in a complex electrical circuit.
Right.
Like if the wire between complex I and complex 3 is frayed, corroded, and dropping the current, methylene blue acts as a brand new, highly conductive copper wire that bypasses the damage entirely and keeps the current flowing to the turbine.
That is exactly what it is doing.
Yeah.
By acting as an alternative electron carrier, it bypasses the sluggish, damaged bottlenecks in the electron transport chain.
Wow.
It restores the flow of electrons, which restores the proton gradient, which maximizes the production of ATP. It fundamentally upgrades the efficiency of the mitochondrial machinery.
So when a biohacker takes methylene blue and reports intense mental clarity, it's not because their brain is flooded with dopamine or adrenaline.
Bro.
It's because the neurons in their prefrontal cortex literally have a surplus of ATP available to maintain sustained activity.
Yes. They have increased their baseline metabolic capacity. The neurons can fire more efficiently, maintain synaptic plasticity, and consolidate memories without depleting their energy reserves.
But wait, I have a question here. By optimizing mitochondrial function, are we just forcing the cellular factory to work overtime, or are we fundamentally upgrading the machinery? Like, does the body eventually become dependent on it?
That is a highly sophisticated question, and it speaks to the principle of homeostasis.
Because you'd think the body would get lazy, right?
You would think so. Does the cell downregulate its own natural complexes because the methylene blue is doing the work for them? However, current research suggests that methylene blue operates cyclically and actually induces a mild hormetic stress.
Hormetic stress. What does that mean in this context?
By stimulating complex 4 directly, it increases oxygen consumption and actually promotes mitochondrial biogenesis.
Biogenesis meaning the creation of new mitochondria.
Exactly. It isn't replacing the natural machinery permanently, it's optimizing the flow, which signals the cell to build more infrastructure to handle the increased metabolic capacity.
Oh, I see.
It is a regenerative signal, not a degenerative crutch.
And that regenerative signal brings us to the next major frontier here.
Yeah.
Longevity and the prevention of cellular aging.
Right.
Because when the electron transport chain is functioning poorly, a lack of ATP isn't the only problem. A bottleneck in the chain produces something highly destructive.
Yes. We have to talk about reactive oxygen species, or ROS.
Free radicals.
Exactly, free radicals. When the electron bucket brigade gets backed up, when there is damage at complex size or complex 3, the electrons don't just sit there politely. They leak out of the chain. And when a high-energy electron leaks into the mitochondrial matrix, it inevitably collides with an oxygen molecule. This collision creates superoxide.
Which is a highly unstable and destructive free radical.
Yes, because that superoxide molecule has an unpaired electron, and the laws of physics dictate that it absolutely must find a pair. It is violently reactive.
So it's like a rogue bouncy ball causing havoc in a china shop.
That's a great image. It will rip an electron from whatever is closest. It will attack the lipid membranes of the mitochondria, causing lipid peroxidation.
Ouch!
It will attack the proteins, causing them to misfold. And most dangerously, it will attack the mitochondrial DNA itself, causing mutations.
Wow. So it's literally destroying the factory from the inside out.
Exactly. This constant microscopic structural damage is what we call oxidative stress.
It is essentially cellular rust. The engine is running, but the exhaust is leaking directly into the chassis and corroding the metal.
Yes.
And the prevailing mitochondrial theory of aging posits that this cumulative oxidative damage is the primary driver of the aging process. It's what causes cells to eventually reach senescence and die.
Our bodies do have natural antioxidant defense systems, enzymes like superoxide dismutase and catalase, and molecules like glutathione that neutralize these free radicals.
Right, our bodies try to fight it.
But as we age, the leakage increases, and our natural defenses are overwhelmed. The cellular rust accumulates.
So how does methylene blue act as an antioxidant? Because we see a lot of products marketed as antioxidants, vitamin C, vitamin E, that just kind of float around waiting to bump into a free radical and neutralize it.
Methylene blue is vastly superior to traditional antioxidants because of its dual action.
Dual action.
First, as we just discussed, it acts as an electron shuttle. By keeping the electrons moving smoothly down the chain and bypassing the bottlenecks, it prevents the electrons from leaking in the first place.
So it's like a net that catches them before they cause damage.
It is stopping the creation of superoxide at the very source. It fixes the leak in the exhaust pipe.
It's prophylactic.
Yes. But secondly, methylene blue undergoes redox cycling. When it accepts an electron, it becomes leucomethylene blue. In this reduced state, it is a potent radical scavenger.
Okay, so if the free radicals are already there...
If there are free radicals already circulating in the matrix, leucomethylene blue will actively donate an electron to neutralize them, oxidizing back into methylene blue in the process.
Oh, wow.
It constantly cycles back and forth between these two states, shuttling electrons and neutralizing free radicals continuously.
Wait, really? It's an auto-reloading antioxidant?
Yes.
Because traditional antioxidants like vitamin C, they donate an electron and then they are spent, right? They have to be recycled by other systems or just excreted.
Exactly.
But methylene blue neutralizes a threat, resets itself, and is immediately ready to neutralize another one or carry another electron down the chain.
That cyclical nature is why it is so highly regarded in longevity research. It is a profoundly effective cellular maintenance tool.
But let me push back a little here. Anti-aging is a massive buzzword right now. Are the sources saying this is a literal fountain of youth? or just a deeply effective cellular maintenance tool?
It's the latter. We have to ground this in reality. Longevity in functional medicine often means extending health span rather than just adding years to a ticking clock.
Health span meaning cellular resilience.
Exactly. By optimizing ATP production and simultaneously suppressing oxidative stress, it protects the structural integrity of the cell. You are delaying cellular senescence.
So if you keep the cellular engine running cleanly, without rust, the vehicle operates at peak performance for a much longer period before the parts inevitably wear out.
Precisely. And by delaying that senescence, you are potentially staving off the macroscopic diseases of aging, which leads us directly into the emerging research surrounding neuroprotection.
Right. Speaking of extending health span and protecting cells from damage, the frontier of methylene blue research is looking at defending us against modern neurological and bacterial threats.
Yes. When we talk about neurodegenerative diseases like Alzheimer's and Parkinson's, we are fundamentally talking about localized cellular failure in the brain.
And the source data points toward methylene blue as a potential neuroprotective agent. How does optimizing the mitochondria translate to protecting against these specific diseases?
Well, in conditions like Alzheimer's, one of the primary hallmarks is the accumulation of misfolded proteins, specifically amyloid beta plaques and tau tangles.
Tau tangles, I've heard of those.
Tau is a protein that normally stabilizes the internal skeleton of a neuron. In Alzheimer's, tau becomes hyperphosphorylated. It detaches, clumps together, and forms dense tangles that physically choke the neuron to death.
Oh, God. And what triggers that tau hyperphosphorylation in the first place?
A massive cascade of events, honestly, but a primary instigator is mitochondrial dysfunction and severe oxidative stress.
Ah, the rust again.
Yes. When the neuron's energy production plummets and free radicals accumulate, the intracellular environment becomes highly toxic, leading to this protein misfolding. Methylene blue approaches this from two angles.
Okay, what's the first angle?
First, by restoring mitochondrial function and suppressing the oxidative stress, it stabilizes the cellular environment, potentially preventing the cascade that leads to tau tangles entirely.
Wow. And the second angle?
Methylene blue has actually been shown in laboratory models to act directly as a tau aggregation inhibitor.
Wait, meaning it stops the tangles from forming.
Its unique chemical structure allows it to interfere with the physical binding of the tau proteins, preventing them from forming those lethal tangles. It is literally dissolving the structural blockages associated with neurodegeneration.
That is an astonishing dual mechanism. I mean, it treats the metabolic root cause of the stress while simultaneously breaking down the physical symptom of the disease.
It is very promising.
But, and I feel like I have to ask this, we are hearing about it fighting neurodegenerative disease and Lyme disease. But the source is clear that more research is needed. Why does science take so long to fully validate these mechanisms?
We must maintain rigorous scientific skepticism here. The mechanisms exist in vitro and in animal models, and early clinical trials are underway. But the human brain is a highly complex ecosystem.
Right, it's not just cells in a petri dish.
Exactly. We have to prove that chronic administration over decades safely translates into clinical preservation of memory and cognition. The rigorous process of scientific validation requires time to fully understand the intricate mechanisms at play.
That makes total sense. And while we are discussing complex clinical challenges, the source highlights another major frontier, antimicrobial properties, specifically regarding antibiotic-resistant infections like Lyme disease.
Yes.
This brings methylene blue full circle back to its origins as an antimalarial, but applying it to modern superbugs.
And the rise of antibiotic resistance is, well, it's one of the most pressing crises in modern medicine right now.
For sure.
Traditional antibiotics typically work by targeting active biological processes in the bacteria, like breaking down the cell wall as the bacteria divides, or blocking the ribosomes from synthesizing proteins.
They rely on the bacteria doing something, being metabolically active.
Exactly. So if the bacteria stops dividing, the antibiotic has nothing to attack.
And bacteria have evolved, right?
Oh, yes. They have evolved a highly sophisticated defense mechanism against this. When exposed to stress or antibiotics, a small subpopulation of the bacteria will enter a state of deep metabolic dormancy.
Or hibernation.
They're called persister cells.
Yeah.
They stop dividing. They shut down their metabolism. They'd essentially play dead.
And because they are playing dead, the standard antibiotics, which only target moving targets, just wash right over them.
Yes. The patient finishes their course of antibiotics, the active bacteria are killed, and the patient feels better.
But then...
A few weeks or months later, those persister cells wake up, turn their metabolism back on, and the infection roars back.
That is terrifying.
It is highly prevalent in complex infections, like tick-borne Lyme disease, caused by the bacteria Borrelia burgdorferi, which is notorious for forming persister cells and hiding in deep tissues.
So how does a 19th century blue dye kill a metabolically dormant superbug?
Because methylene blue does not rely on active cell vision to exert its effect. Remember, it interacts directly with electron transport and cellular metabolism.
Oh.
Emerging research suggests that methylene blue can penetrate the bacterial membrane and interact with the dormant metabolic machinery.
So it bypasses the normal defenses.
Exactly. Depending on the exact concentration and cellular environment, it can force the persister cell's metabolism to reactivate, essentially waking it up so that standard antibiotics can destroy it, or, alternatively, at higher intracellular concentrations within the bacteria, it can generate a massive, localized burst of oxidative stress.
Wait, I thought it was an antioxidant.
It is, in our cells, at low doses. But inside the bacteria at high doses, it literally weaponizes reactive oxygen species to destroy the persister cell from the inside out.
It's an entirely different vector of attack. It bypasses the traditional targets like the cell wall and goes straight for the mitochondrial equivalent within the bacteria.
It is a profound shift in pharmacological strategy. By targeting fundamental electron transport, it sidesteps the standard mechanisms of antibiotic resistance.
Okay, we have outlined an incredibly powerful molecule. Because these mechanisms are so powerful, this isn't something you can just casually experiment with.
Absolutely not.
It bypasses the blood-brain barrier, it rewires mitochondrial electron transport, it scavenges free radicals, it alters protein aggregation, and it disrupts dormant bacteria.
Yes.
When a molecule has that level of systemic, profound biological influence, it commands absolute respect. You cannot casually experiment with something that manipulates your foundational energy current.
Yeah, you absolutely cannot. The potency of methylene blue is a double-edged sword. Manipulating the mitochondrial matrix requires exact precision.
Which brings us to the strict guardrails, dosing parameters, and absolute medical contraindications associated with its use.
Right. Knowledge is only valuable when applied safely.
The functional medicine protocols are very explicit. This must be overseen by a licensed physician. It is not a DIY supplement. And the dosing is the first critical guardrail. The standard metric utilized in clinical settings is remarkably low, typically around 1 milligram of methylene blue per kilogram of body weight.
That specific dosage is derived from the concept of hormesis, which we touched on earlier.
Right.
Hormesis describes a biphasic dose-response curve. At a low dose, methylene blue acts as an electron donor and an antioxidant, optimizing ATP production. But if you massively exceed that dose, the molecule's behavior fundamentally flips.
It stops being a helper and starts becoming a hindrance.
Worse, much worse. At highly concentrated doses, instead of donating electrons to cytochrome C, it starts stripping electrons away from the natural machinery.
Oh, wow.
It becomes a powerful pro-oxidant. It will actually induce severe oxidative stress and halt ATP production.
The dose literally dictates the poison.
Yes. This is why self-titration without medical guidance is deeply irresponsible. Biological systems require precision.
And even at the correct low dose, there are severe, non-negotiable medical contraindications, right? I mean, we really need to highlight this. The most critical risk involves a pharmacological interaction with psychiatric medications, specifically SSRIs, selective serotonin reuptake inhibitors.
Yes, this is a life-threatening interaction known as serotonin syndrome.
And to understand why this happens, we have to look at an enzyme in the brain called monoamine oxidase, or MAO.
Right.
MAO is the enzyme responsible for cleaning up neurotransmitters, right? Like after serotonin has done its job transmitting a signal between neurons, MAO comes in and breaks it down to keep the system balanced.
Correct. Specifically, MAOA breaks down serotonin. Now, methylene blue, purely by chemical coincidence, is a highly potent reversible inhibitor of MAOA.
Meaning it stops it from working.
Yes. When you take methylene blue, you are temporarily paralyzing the cleanup crew. The serotonin is not being broken down.
Under normal circumstances, maybe that just results in a slight elevation of mood. But if someone is on an SSRI...
If someone is on an SSRI, they are already actively preventing the reuptake of serotonin. The SSRI is forcing the serotonin to stay in the synaptic cleft.
Okay.
If you combine that with methylene blue, which prevents the breakdown of the serotonin, you create a catastrophic bottleneck. The serotonin levels in the brain skyrocket exponentially.
And the brain cannot handle that excess. Serotonin syndrome leads to hyperthermia, severe agitation, tremors, delirium, and in severe cases, it is fatal.
Yes. It is an absolute hard stop.
Yeah.
Methylene blue cannot be combined with serotonergic drugs.
It is a prime example of why understanding the deep biochemistry is necessary. The molecule doesn't just do one thing, it interacts with multiple enzymatic pathways simultaneously.
Exactly. And that leads to the second major genetic contraindication. Individuals with a specific genetic anomaly called G6PD deficiency must avoid methylene blue entirely.
G6PD stands for glucose 6 phosphate dehydrogenase, right? This is an enzyme that helps red blood cells function normally. Why does lacking this specific enzyme turn methylene blue into a danger?
We have to circle all the way back to how methylene blue acts as an antioxidant. Remember, to neutralize free radicals, methylene blue must first be reduced into leucomethylene blue.
Right.
To achieve that reduction, it requires an energy molecule called NADPH to donate an electron.
Right. NADPH is the fuel that reloads the methylene blue.
Exactly. And G6PD is the primary enzyme in the red blood cell responsible for manufacturing NADPH. It operates in something called the pentose phosphate pathway.
Okay, so if a patient has G6PD deficiency, their red blood cells cannot produce enough NADPH.
Right. So if they take methylene blue, the methylene blue enters the red blood cell, but there is no NADPH to reduce it. It gets stuck in its oxidized state.
Oh, and stuck in its highly concentrated oxidized state, it acts as an extreme pro-oxidant.
Yes. It begins tearing electrons away from the lipid membranes of the red blood cell itself. The red blood cell literally undergoes oxidative rupture. It explodes.
Wow.
This triggers a massive systemic destruction of red blood cells known as hemolytic anemia.
That's terrible. It takes the very mechanism designed to protect the cell from oxidative stress and reverses it, causing catastrophic oxidative damage.
Yes.
Again, this highlights why this cannot be a casual biohack. A simple genetic nuance changes the entire chemical interaction.
Which is why thorough blood work and genetic screening are the absolute foundation of any legitimate functional medicine protocol involving potent compounds.
Yeah.
Personalized, physician-led medicine is so critical.
And the final critical guardrail involves the sourcing of the molecule itself. We mentioned at the very beginning that methylene blue was originally created as a textile dye, and it is still heavily used today in industrial chemistry, aquaculture, and laboratory staining.
Right.
But the source is adamant that only pharmaceutical-grade or USP-grade methylene blue should ever be ingested.
This is a matter of profound toxicity. When chemical manufacturers synthesize methylene blue for industrial or laboratory use, the process involves heavy metals.
So it's not clean.
No, industrial-grade methylene blue is frequently contaminated with significant levels of arsenic, lead, and cadmium.
Because if you are just dyeing a pair of jeans or, staining a slide for a microscope, trace amounts of lead or arsenic do not matter.
Exactly.
You wouldn't put dirty, unrefined fuel in a high-performance race car. That's why pharmaceutical grade is the only option here. The manufacturing process isn't designed to filter those metals out because there is no biological safety requirement for fabric.
Precisely. The purification process to remove those heavy metals and achieve USP pharmaceutical grade is extensive and expensive.
So if someone tries to cut corners,
If someone attempts to biohack on the cheap by purchasing chemical-grade methylene blue from a laboratory supply company, they are directly ingesting highly toxic heavy metals.
it's the ultimate biological irony. You are taking a compound to theoretically protect your mitochondria from oxidative stress, and instead you are bombarding your mitochondria with arsenic and lead, which will permanently destroy the cellular machinery and cause severe neurological damage.
It completely defeats the entire purpose of cellular optimization. Purity is non-negotiable.
Let's step back and just look at the massive expanse of territory we've covered today. We traced the incredible arc of methylene blue from its synthesis in a 19th century German chemistry lab as a simple textile dye to its revolutionary use by Paul Ehrlich as the world's first synthetic antimalarial drug.
We examined its critical role in emergency medicine, where it chemically unwelds the molecular doors of hemoglobin, reducing ferric iron to ferrous iron to rescue tissues from cellular hypoxia.
And we followed its path across the highly selective blood-brain barrier, discovering how its lipophilic nature allows it to bypass security and act as a profound nootropic.
Right.
We unpacked the deep biochemistry of that process, how it physically acts as a parallel wire in the mitochondrial electron transport chain, shuttling electrons to complex 4, maximizing the ATP production, and simultaneously neutralizing destructive free radicals at their source.
We discussed its potential to disrupt the physical tangles of neurodegenerative disease and wake up dormant, antibiotic-resistant superbugs.
But we also balance that potential against the absolute necessity of rigorous medical oversight,
Yeah,
acknowledging the severe risks of serotonin syndrome and hemolytic anemia. It is a molecule of incredible contradictions and profound power.
it really is.
It is a brilliant reminder to you, the listener, that the quest for optimal health, the desire to extend our health span and upgrade our baseline metabolic capacity, is complex and deeply personal. It's not about finding a magic consequence-free pill.
No, true optimization is an exercise in biological precision. It requires a deep, respectful understanding of our own microscopic machinery.
And it leaves us with a fascinating, lingering thought. We are currently utilizing the absolute cutting edge of modern biochemistry, advanced spectroscopy, genetic screening, and molecular mapping to understand a molecule that was synthesized in 1876.
Yeah.
A molecule that was sitting in plain sight, coloring fabric, while the science of human biology slowly caught up to its potential. It makes you wonder, considering methylene blue was the very first synthetic drug created back then, what other forgotten, obscure compounds from the 19th century might be sitting in dusty archives or industrial catalogs right now?
It is a compelling question. The archives of early chemistry might hold the blueprints for the next revolution in longevity.
What other molecules are just waiting in plain sight for our 21st century understanding of cellular biology to finally unlock their true, untapped potential?